Today I'm going to tell you a little bit about the Single Cell Portal, which is a web-based data portal for sharing and exploring single cell genomics data. And I'm going to do this in two parts. First, I'm going to show you some slides where I'll give you some background and walk you through some of the features of SCP that I think are the most cool and interesting. And then I'll also take you through a hands-on demo so that you can see how these features work in real life. So at this point, you might be wondering, what is the Single Cell Portal? As I said before, it's a web-based data portal that you can use to explore and share single cell genomics data. But more than that, it is part of its value is that it's a centralized and standardized repository for this kind of data, which makes it a lot easier for people to discover your data and to work with it and analyze it if they want to, if it's interesting to them. In addition, it's designed to be a place where you can explore these data without requiring any computational experience whatsoever. Instead, we have a suite of dynamic, interactable visualizations that you can use to try to understand the patterns in these multidimensional data. And finally, SCP is designed to support all phases of the research lifecycle, from when you've just collected data and are sharing it with collaborators to try to suss out what the story, what story those data tell, to once you've figured out what that story is and you want to share it with the broader research community. And now is a really exciting time to be working with SCP because we're increasingly getting more and more data and now have a really rich set that come from researchers all over the world. I think that you guys might find that there's a lot of value to accessing that richness, which, both gives you a broad audience for sharing your data and gives you the opportunity to discover some really cool data that are hard to find when you're just looking for them piecemeal. So as I said, I'm going to take you through a sort of highlights reel of the things that I think are the most useful and interesting when you're just starting to use the portal. So the first one is, once you get to the portal, you have to be able to identify the data that you actually want to look at. And for this, we have a few different kinds of search options, which I'll show you more in the demo. There's a basic search where you just type in some text, such as a disease or an organ, and that will search through the descriptions for all the studies on the site and return any studies that mention that term that you search for. There's also a more advanced faceted search that allows you to filter these results down based off of specific facets, such as cell type or anatomy. And this just makes it a little bit easier to narrow your search results so that it's more sort of easy to wrangle all of those results. And in addition to being able to search across studies based off of this kind of terminology, you can also search for individual genes across studies. And I'll show you more about that in the demo as well. Next, perhaps the coolest part of SCP is the large suite of dynamic visualization tools that we offer to help you explore the data. So I'm going to take you through a few of these, though there are many different options, and I invite you to explore SCP to try to discover them all. So one really common kind of plot that you'll see for many studies is a scatter plot like this. This is the result of a dimensionality reduction that's been applied to a full data set, where that data set contains measured expression for a large set of genes in a large set of cells. So it's very high dimensional, but this has been projected into 2 dimensions in order to see the major patterns and the major axes. So in this kind of plot, each point corresponds to a cell or a bead, for example, depending upon how the data have been measured. And these points are all colored according to some sort of metadata that are included in the data, such as cell type or whether the sample came from a healthy organism or a diseased organism. And so, for example, you can explore different kinds of patterns by changing these labels around. And in some cases, the data are 3-dimensional, which is a really big benefit to be able to interact with 3-dimensional plots on the portal rather than in a manuscript, for example, because you can really appreciate the richness of the shape of the data when you can interact with it. Another really exciting and new type of visualization that we have is a way to look at spatial transcriptomics. So for example, imagine that you've taken a slice of tissue like this, and you've put a matrix of beads on top of that slice in order to measure genetic expression at every location within the slice. Then once you've done that or measured something like cell type, you can then create a plot like this, which shows you it maps some information of the data, such as cell type, to every location on that slide and helps you understand the spatial distribution of the data. So when you look at this alongside a plot, another dimensionality reduction plot like this that might be colored according to clusters that exist in the data, you could see interesting trends both in the data's expression values and in space. So for example, you might notice that this purple cluster is very separated in genetic expression space from the rest of the data. And it's also relatively segmented in anatomical spatial space, which might suggest something interesting about the mechanism behind that group of cells. I'll go through the remaining visualizations with less detail. We also have some violin plots that allow you to see the distribution of different genes expression in different segments of the data. There are also dot plots and heat maps that help you understand how different genes are related to each other in the data. There's also an option to create and save sort of annotations within the plots so that you can identify interesting subsets and share them. And finally, we have an ideogram viewer that helps you to search for an individual gene within a given study, and then quickly identify other genes that might be related to that gene, which might help you figure out your next target. So once you've been able to explore other people's visualizations, you might want to create your own study. And when doing so, you might want to be able to control who accesses your data and when. And so in anticipation of this, we tried to give you a lot of control over this aspect of the study. So first, SCP is included in Terra's security boundary, meaning that it's BICCN moderate, which gives it a layer of security. But even beyond this, we make it possible for you to control who gets to see your data based off of whether the study is a public study or a private study. In addition, when you share a private study or when you share any study with individual collaborators, you can control whether those collaborators can edit the study or just view it. And another really cool feature that we've just been working on is an easy to use system for anonymous reviewer access to your data. So if your study is private because you don't yet want to release it to the world until you've published it in a journal, but you want to be able to give access to the study to reviewers so that they can play around with the data themselves. You can use this system to basically send a pin to the journal and they'll pass that along to the reviewers, and then you can control how long they have access to your data, and they'll only be able to view your data without actually downloading it. But I'll tell you more about that in the demo as well. Another really cool feature of SCP, piggybacking on Liz's great presentation about running analyses in Terra, is that SCP is integrated with Terra, as I mentioned before. So you could put some data in Terra, run some batch analysis pipelines, or write some customized analyses using your notebooks that might produce some standardized visualizations. And then you can publish those data and those results to SCP as a study. And then if you decided to sort of change your analysis along the way to create new results, you could sync those data between Terra and SCP. It just kind of reduces the barriers to sharing and analyzing data. Another way that we've tried to do this, which I won't go into as deeply, is to set up a few different ways to use scripts and APIs to access SCP's functionality, for example, to upload your data or to sync it. There's more documentation about this on the SCP website if you're interested. And finally, we've recently set up a system called Study Collections These are kind of curated sets of studies within SCP that are united based off of the research consortium that's working on them or the particular research topic. So for example, right now, we have a collection for the BICCN Anatomy and Morphology project, which is all of these studies are also available on the wider SCP, but it just allows you to sort of search through a subset that might be particularly useful for people that you collaborate with or with whom you want to share your work. We have another collection for COVID studies that were recently released. So as promised, that was a somewhat whirlwind tour of some of the highlights of SCP's features. I just want to mention before I transition to the next part of this presentation that I'm deeply grateful for the rest of my team, which is a dynamic group of designers, software engineers, and computational biologists, and to our funders for making this work possible. I'm going to throw up a few links to different resources